we are going to see about the blender experiment. It is known as blender experiment and also Hershey and Chase experiment because the scientists Hershey and Chase are going to propose this experiment. Alfred Hershey and Martha Chase are going to propose this experiment, therefore it is known as Hershey and Chase experiment. It was known to be blender experiment because a kitchen equipment blender is going to be used in this experiment, therefore it is known as blender experiment. A main principle in Griffith and Oswald Avery McLeod experiment is going to be pneumonia causing bacteria, that is R and S strain. Here a main component to be used is going to be E. coli and phage T2 bacteria, phage T2 virus, bacteriophage, and the E. coli bacteria are going to be used to identify, which is going to provide a evidence to identify DNA as a genetic material. It is going to be a confirmation experiment. So, Griffith experiment and Avery McLeod experiment only provide the transformation factor to identify the transformation factor, but blender experiment is going to provide the confirmation to find DNA as a genetic material. So, here a kitchen wearing blender is used. It is known as blender experiment. First, we should know about the bacteriophage to know about this experiment. So, a bacteriophage is going to be a virus. This virus is going to affect the E. coli bacteria in general. Like a Rutstein bacteria and Yerstein bacteria are pathogenic to mice. Here, instead of bacteria, virus is used, which is bacteriophage, which can affect E. coli only. So, a bacteriophage can inject its genetic material. Until that it was not found DNA as a genetic material. So, here we can find that a genetic material is injected by the phage into a bacteria, which lead to progeny-phage production and lead to death of the bacteria. So, here this principle is going to be used. Here we are going to use the T2 phage, which is going to be a virus. T2 bacteriophage is going to be used against the E. coli bacteria. A bacteriophage here is encased with its genetic material, that is DNA, and protein is going to be a shell behind it. Here we can find the structure of bacteriophage, T2 bacteriophage, which can cause a pathogenicity to the E. coli bacteria. It is a protein core. It is going to be DNA. It is going to be a collar. It is a tail fiber. So, when the bacteriophage, so a protein core is going to be outside the E. coli. Only the genetic material which is going to be transformed or transferred into the E. coli. After that, as it was a virus, a bacteriophage is also a virus. So, it hijacked the E. coli nucleus system and produces more progeny. This progeny are going to be released, which causes the death of E. coli. It is a main cycle of bacteriophage in E. coli. So, based on this, we are going to find DNA's genetic material, which is going to provide a evidence by blender experiment. So, here they use a principle of radioactivity labeling. As we said that a protein coat is going to be outside and DNA is going to be inside. So, a DNA in general consists of ribose sugar and the nitrogenous base. These nitrogenous base and ribose sugar are going to be connected by glycosidic linkage, and this ribose sugar is going to connected with the another ribose by phosphodiester linkage. So a DNA consists of phosphate which provides the phosphodiester linkage. So the phosphate is only unique thing in DNA. Likewise, a protein consists of sulfur containing component is going to be a protein. So here we are going to label the phosphate 32 into the DNA phosphate and sulfur 32 into the sulfur of protein. So that a DNA does not have sulfur and a protein does not have a phosphate is the main principle. So the DNA is going to be labeled with phosphate. As the protein shell has methionine and cysteine, the sulfur is going to be labeled. So a phosphate 32 is not in the protein. Therefore, a phosphate 32 is labeled in DNA and a sulfur 35 is going to be labeled in the protein. This is going to provide a difference and providing the image representation and give a evidence to know the DNA as a genetic material. So the labeling difference of DNA and RNA can be found out because the RNA is going to be depicted the protein. So a DNA is phosphate 32 labeled and the protein is going to be sulfur 35 labeled. So now this labeled bacteriophage is going to be allowed to infect the E. coli. When it was infected off, after the time it infected, it was agitated with the blender. It was mixed with the blender. After shaking, it was centrifuged. When it was centrifuged, it was found that a radioactive phosphate 32 is going to be associated with bacterial cell, and whereas S35 is on surrounding medium, which means I have already told that there is a bacteria. The phage is going to inject its genetic material into its bacteria, which can hijack the nucleus system of bacteria and produces more progeny. So here we can find only that genetic material is going to be in. No proteins are going to be in. So when it was agitated off, the genetic material found associated with the E. coli can be found as phosphate 32 label, whereas a surrounding medium consists of protein shell, which means a protein shell without any genetic material are found to be on the surrounding medium. So a genetic material which is transferred is going to be only phosphate 32 labeled DNA. Thus it was found. So the phage progeny studied for radioactivity has phosphate 32 labeled and not labeled with S35. It confirmed that a transformed material is DNA, not a protein. Therefore, it was found that DNA enters into the host cell is going to transfer the genetic information. Thus, a DNA as a genetic material was found. Here we can find the image how the experiment is carried out. So S35 labeled and phosphate 32 labeled bacterial culture are going to be allowed to infect the, it was S, phosphate 32 means DNA labeling, S35 means protein labeling. Both labeled components are allowed to infect the bacteria E. coli. So the T2 bacteriophage is going to infect it, which is labeled. When it was blended with kitchen blender and it was undergoes centrifugation, it was found that S35 labeled are found to be on the top and phosphate 32 labeled are found to be bottom. Phosphate 32 labeled are found to be bottom because it is associated with cell. When it was associated with cell, it has more density, therefore it was sedimented at the bottom. Where it was without cell, therefore it has less density, it was found to be at the top. Therefore, the radioactivity labeling show that a cell, bacterial cell is associated with phosphate 35 labeled, 32 labeled component, that is DNA. Thus, a DNA as a genetic material was found.